Now it's time to welcome Tiina Roose. She holds a chair of biological and environmental modeling at the School of Engineering at the University of Southampton. In 2023, she was appointed the deputy head of the School of Research, one of the largest and most research-intensive engineering schools in UK, engaging in research across a wide spectrum of engineering disciplines, from very small scales to very large scales like ship, aerospace, and satellites. Tiina was born in Estonia under the Russian occupation. She completed her undergrad degree in control system engineering at the Tallinn Technical University. And following this, when Estonia became independent and borders opened, she did, after a short stint as an investment banker, MC and Doctor of Philosophy in Applied Mathematics at the University of Oxford. A specialty with a graduate student was to develop a multi-scale mathematical modeling techniques to answer scientific questions about how the biological branching structure, such as plant root, interacts with their environment. And following the completion of her Doctor of Philosophy in 2000, she moved to the Harvard Medical School in Massachusetts and Massachusetts General Hospital as a postdoctoral fellow to work on mathematical modeling of lymphatic function during cancer development. Upon her return to Oxford in 2003, she was awarded a Royal Society University Research Fellowship to work on modeling of plant roots, lymphatic blood vessel, and lung physiology. She held her URF first in Oxford, and upon the birth of her son, she moved to University of Southampton in 2009. In 2013, she was promoted to professor, and after the completion of her Royal Society URF in the same year, she was awarded an European Research Council Consolidation Grant on data-intensive modeling of rhizosphere processes. I hope I pronounced rhizosphere correctly. In 2019, she was appointed Royal Society Wollstone Fellowship at Southampton. Tiina is the chair of the Faculty of Engineering Environmental Fellowship, academic, mentoring early career researcher, and also serves on the UKRI Transformative Technological Strategic Advisory Panel. Tiina, the floor is yours. I stop sharing. You're welcome to start your talk.
Hello, it's a real pleasure to be here with you. And the sharing is not working.
Was working before. It's always the famous.
Try it again.
Murphy's law is inevitable.
Hello.
Okay.
So
see it.
Yes, it's a real pleasure to be here, give a talk to such an international audience. And thank you for your introduction to this talk. So I am a modeler. My training was applied mathematics, but now I work in engineering department. And so in engineering, we tend to always try and think of why is it that we're doing things? Are we doing anything useful? While some applied mathematicians do it, I want to now tell you why I'm doing this image-based modeling of plant-soil interaction. So my motivation always has been to contribute my knowledge and skills towards growing more food for humans. And that all human food effectively is underlined by plants. So even meat production requires plant products as a feed into cows and animals. So how do we grow the crops? Much of in 20th century, the crop yields increased because of heavy fertilization and fertilizers that came available. Now the problem is that rich countries use too much fertilizers in soils, and that leads to pollution of waterways and seas. Poor countries, on the other hand, can't afford these fertilizers. Now, lots of our work has been done to do with phosphorus fertilizers, because those models, when I started working in this field, were spectacularly inaccurate. If they got within 30% of explaining field trial results mechanistically, they were considered good models. So we started looking at phosphorus models, but because the phosphorus is also now predicted to run out in the world in the next 50 to 100 years, depending on which expert you believe. The rock mineral resources are limited. And until then, when they run out, the major source, the places where they get mined are Morocco, Syria, China. And so whilst we can think of alternatives to rock mineral phosphorus resources in the form of human digestate and extracting new fertilizers from sewage works, these fertilizers and new fertilizer products will be more expensive. So we do need to find alternative ways to heavy fertilizations of our crop, of our farm fields, in order to still grow the crops. Now, there are two different sides to this. We can deal with plant traits. We can breed plants. So, controversial subject in Europe is genetically modified plants. So we can breed plants or modify plants to be more effective at taking up nutrients naturally from soil. But we can also do the soil amendments. So the problem here is that which one should you be doing? And obviously the field trials, running lots of field trials, is very expensive and also time consuming. It takes time and effort to do this. So my group's approach has been to develop precision mathematical models so that we can then run these models in a predictive manner to find the strategies in silico in a computer, so that we can optimize the use of field trials and do the precision guiding of the experimental approaches. But the big problem with mathematical modeling of plant-soil interaction is that we can't see into the soil. The soil is opaque, and the most obvious approach to imaging or what's happening in soil is using imaging techniques like X-ray CT. I don't know what this audience knows about the X-ray CT, but here's a little example of the how X-ray computer tomography works. It basically rotates the sample in the X-ray beam, through lots of angles, taking lots of pictures, which gives you basically a density map of what you're seeing, and so you can see into quite dense materials like soil. This allows you then to segment out the structures of, in his case, you see how plant root is interacting with soil particles. But at that point, when you've done the imaging and segmentation, you still end up with pretty pictures only. So you can see what's there, but you don't know how that thing is functioning. And what the image-based modeling approach does is it integrates using mathematics and computation the function into this into the imaging loop, and then using this functional characterization of the structures, we can plan the next experiment. The other way of looking at image-based modeling workstream is to look at it from the point of view of fast computational methods and slow computational methods. So if we image and segment, we get our pretty pictures. We can take it both ways. We can take the geometry that we observe mesh it as it is using computational techniques, and then run the simulations on those images directly. Now, whilst the computer power is increasing, this process is still quite slow, and it takes and you can't do it on lots and lots and lots of images because we're dealing with 3D complex volumes. But it's obvious that from imaging we can also take fast morphology calculations that are based on sort of the measures of tortuosity of a network or feature you're observing, the volume fractions, things like that. The challenge with all these fast morphology approaches is where you're extracting statistics from the images, that you don't know which ones of the statistics, what statistical measures of the image correlate or are the greatest indicator of the function. And so doing both approaches simultaneously allows us to basically do this structure function association to enable the fast throughput of imaging processes. But what is the main challenge for modeling soil and soil plant interaction? The main challenge is that the problem is massively multi-scale. So we have things that are on a molecular scale happening when it comes to fertilizer dynamics, plant function, and then we move through to cellular scale, organism scale, field scale, and ecosystem scale, because eventually we want to use the models to manage things, manage crops on regional scales, ecosystem scales. How should we preserve and allow for carbon deposition in soil? And so this is where this loop of multi-scale, field scale experiments to modeling to then back to experiments is becoming really, really important. And the key really is linking all of these things up. The other way of looking at this is to carry the experiments and models together. It was quite clear, sort of now, I guess 15 years ago. So modelers, we mathematicians, this is SciAm after all, have been working for a long, long time in multi-scale analysis and developing mathematical and computational techniques to move across the scales. Whether you're working on, you use different techniques when you're working on soil particle scale, column scale, single plant root, your combination of analytic tools with numerics. We mathematicians are built to do it. But roughly 15 years ago, it became clear that this translation of modeling across the scales is only ever going to be as good as the calibration of each models at each scale, so that we will also double check and validate the models on each of these scales. And so we started in Southampton when I arrived in the engineering department, where we had lots of different experimental techniques and capabilities to do this thing where we calibrated models at each scale. And as you notice here, the experimental techniques as you move across the scales will have to change, because X-ray computed tomography, for example, can't be applied on a field scale. The machines just don't, transport for this purpose. And so X-ray techniques tend to be on smaller scales. And as you move up, it becomes more important to validate the models using the novel tracers or sensors and other sensing techniques. So we started doing this, and I will give you a couple of easy sort of illustrative examples where this has worked really well in combination with mathematical modeling. So the first example I'm giving is the root hairs. So all roots, most wild-type plants in the world, whether it's crop plants or plants in natural ecosystems, have hairs. So some roots are hairier than the others. Plant breeders now can breed wheat plants or barley plants and rice plants that are more hairy or completely bald. The question is, it's been hypothesized that these hairs, which are unicellular extensions of the root surface epidermal cells, are very, very important for uptake of these strongly bound nutrients like phosphorus, because it's intuitively clear they might go into pore spaces and take up more locally the phosphorus that way. But what do we want? Do we want the plant breeders to breed longer hairs with lower density, or higher density hairs but shorter? Do we want to overexpress transporters for phosphorus on root hair surfaces? These questions were all open for a debate. So one of the first studies we did was to develop models for these hair functions. And we used the published data on our root morphology. So we used standard toolbox in mathematical for applied mathematician, and that is, let's assume that the root hairs are this idealized regular geometry, and this allows us now to use matched asymptotic expansions and homogenization theory to develop a model that describes how these root hairs take up phosphorus and what their effect is. So assuming that root hairs are perfectly parallel cylinders on a root surface, and that in terms of number, they number over length scale is a lot higher than they size. We started with homogenization theory. So to just quickly maybe recap, I know you're all familiar with this. How the homogenization works is it assumes that there is a macro scale, spatial scale, which is large, and then there's microstructure that is small. And what the starting point for this technique is always you write down the equation, which in this case is diffusion equation, that is valid on all scales. But then we find, using asymptotic expansions, the solution to it that depends independently on that micro scale and macro scale variable. So by virtue, we then also expand all the spatial derivatives in these variables and collect together the orders of problems that we get in different orders of epsilon, that small variable that is a ratio of microscale to macroscale variable. So again, this is fairly standard sort of multi-scale analysis of diffusion equation. Now, what it came to when we were dealing with a root hair problem is that when we started validating the model based on the published experimental data on root hair morphology, we found that there are three different root hair effective models that we can derive. The first one was negligible hairs. It's basically what it says, that the root hairs, they don't take up enough very strongly the nutrients. They sit there as sponges in a space, but they don't generate a significant sink term for the nutrient movement in the macro scale diffusion equation. The next model was where we do have a diffusion, a sink term present. So basically the diffusion, effective diffusion in between the root hairs occurs roughly on the same time scales as the root hair phosphorus uptake. And the third one was dominant root hairs. So that is the limit of where root hairs basically sort of go into the pore space, the soil pore space, and suck up everything really fast, leaving nothing behind. Now, when we looked at the data, what we found is that all the in vitro model, all the experimental data pointed that it's these little hoovers going into pore space, so that the dominant root hairs model was the most likely candidate to be characterizing what the root hairs were doing, with only some indication that it's the dynamic hairs. But the problem with that was that all the root hair morphological studies were only ever done in the gels in the lab, because you can't see what the root hair density, what the root hair morphology in soil is, because it's opaque. And even if you try and wash out you'll lose hairs. So we basically decided we need an imaging technique to really observe the root hairs in soil, and to really settle off which one of these models is the correct one, the possible 3 candidate models for root hair phosphorus uptake. And so at that point, it came that we need to go and image. Now, the technique that allows for this imaging is the X-ray computed tomography. The problem is that whilst these root hairs are 10 microns across and 1 millimeter, up to 1 millimeter long, so our spot size that we need to image these features has to be of the order 1 micron. Now, tabletop CT scanners can achieve that resolution, but that comes at the expense of scan time, which can be for that resolution in tabletop system of the order of 8 hours. But roots are actually grow quite a lot. They're quite active in soil. So plant roots grow 2 centimeters per day. So tabletop systems weren't suitable, because during that time plant roots would be growing. So we developed these assays to image these hairs in the synchrotron beamlines, where the scan times are a lot faster. Thus, we can not only do high resolution imaging of root hairs, but also high throughput imaging, so we get replicates and things. And you would have seen here in a movie now how we have to build a bespoke assay to enable to do this imaging at these fabulous synchrotron facilities. So, cut the long story short, we took these images, we segmented out the root surface, root hair surface, and soil pore space, and then used these geometries to do the calculation of the phosphorus movement in this soil pore space. So what we have is, on the root and root hair surfaces, we have the Michaels-Menten type nutrient uptake law, which basically says that when the nutrient concentration in a pore space is very low, the uptake by hairs and root surface increases linearly, but after a certain concentration, it plateaus off, and that there's a maximum capacity for the rate at which the root surfaces can take up phosphorus. On the particle surfaces, we had the first order binding reactions, because the phosphorus can attach to particle surfaces and then get released into pore space as the pore space concentration reduces. So accounting for all of it, our results basically showed that for different phosphorus indexes in soil, and so this is UK thing, but US has its own equivalence of it, that the phosphate index 1, 2, and 3, which basically translates more or less phosphorus in soil, for all the cases, the presence of root hairs roughly doubles the phosphorus uptake. So that now indicated that it is actually the balanced diffusion pathway, that they are not doing root hairs. The effective model to translate the root hairs from our homogenization work is that sync term model, the balanced uptake, not the little hoovers in a pore space model, that they are balanced with the root. Image-based model also in this case allows us to now quantify how local is this phosphorus uptake. It's intuitively obvious that as the root hairs extend into pore space, they can take up more locally. But modeling and doing it on image-based basis allows us to quantify what the metric, how much more local it is. And in this case, in the case of these roots, is that the root hairs extract roughly about half the locality compared to the root, which extracts the phosphorus from a much wider soil particle surface area. So this was useful for us in a sense that we now decided, okay, we know what model to carry through to the field scale models to account for root hairs. Remember, these plant breeders can breed for hairy or bold roots and everything in between. So this inspired us to take modeling, and why is it not working, to deal with the issue of mycorrhizae. So mycorrhizae are fungi that grow in soil that you've heard can communicate between the trees, and from one tree can talk to another using these fungal networks that link their roots. So all crop plants also have mycorrhizal symbiosis, and equally they are thought to be super important for uptake of nutrients from further away. So if the plant is in a phosphorus-starved situation locally where their root system is, it can build up this symbiotic relationship with mycorrhizal fungi in soil, which then allows them to get where fungus brings the phosphorus from quite far away. It's could be meters, kilometers in some cases, and then exchange it for carbon, because mycorrhizal fungus needs to grow in the soil. It needs carbon somewhere, and because it doesn't have a above ground bit, it needs to get it in exchange from the plant. So it's a trade-off. Plant gives carbon to fungus, fungus gives nutrients to plant. So those features of the mycorrhizae, hyphae that do the work, are also roughly the same size as root hairs. It's just they're a lot longer. They form a network. And they form this network by having a tip, growing tip. So the organ that grows in a mycorrhizal fungus is the tip. And this tip can split into now two little tips. Those tips can also be destroyed in a sense of when they form a loop. So two tips can meet and have tip-tip anastomosis by forming, and that forms a loop. Similarly, the tip can fuse with the other hyphae that forms a loop. So we developed the first model for this mycorrhizal network development, and we did it by tracing the tip movement. So it's a tip flux N that is moving in space, and the trail that it leaves behind is now the hyphae in the soil row. And this tip splitting and tip side branching and anastomosis is counted as a birth and death effect for the new tips. Integrating this model that predicts hyphal length density effectively with a phosphorus uptake model now allows us to predict how the phosphorus profiles in the soil around the roots develop. But again, the problem was the same, like with the hairs, is that whilst you can observe the hyphal distribution growth and evolution in time. So we can develop this, we can validate the model for fungal growth really well. There was quite a lot of good data for that, to actually image what happens to the phosphorus, that are they sucking up all the phosphorus behind or not, was really lacking. And so we started doing that, okay, we could see the root hairs. So we should be able to, with X-ray CT, we should be able to see the root mycorrhizal fungi with the same technique. Now, the caveat is that you need to grow both plant and fungus in this case. And being in an engineering department, we developed these quite sophisticated engineering assays to enable the imaging of mycorrhizae. So what we have here is, if you see, there's a plant compartment, and then these little syringe barrels on the side here are the fungal compartments. And they both linked, but what we have is that there is a mesh so that the roots can't grow into little syringe barrels, only fungi can. And this way we can grow them. We have them in bespoke box. We can fly them to different synchrotron light sources across the world, and then image how the evolution of hyphal density and the phosphorus profiles in the soil in these little side branches. And what we found is, so we were able to do, courtesy of the ERC funding, quite a thorough first study of this, where we had a control soil which wasn't inoculated with mycorrhizal fungus, and then soil that did have mycorrhizal rhizophagus inoculation in it. And what you see is that from images, we already, I hope you can see, is that the control sample, which doesn't have mycorrhizal fungus in it, has the native saprophytic fungi in soil. They morphologically quite different. Whereas the soil where we imaged, which was inoculated, has a lot more regular, straight sort of mycorrhizal fungi in it. So we integrated the inoculation treatments, but we also put in the phosphorus treatment. So some of the syringes we did add fertilizer granule, in the others we didn't. And now by correlating the X-ray computer tomography with X-ray fluorescence elemental mapping for phosphorus on the same samples, we could validate the mathematical model for the mycorrhizal phosphorus uptake to answer the questions that were really critical for us. And the question was, what is the rate of mycelial uptake? And what we found is that it allowed us to narrow down the rate of mycorrhizal phosphorus uptake by quite many orders of magnitude. And here I show you how that actually varied, that rate of uptake, depending on whether the soil was fertilized or not. So IP is inoculated, added phosphorus treatment, and IP minus is without phosphorus treatment. And the difference is really now, it showed the first proof of principle of what can be done with techniques, X-ray techniques for the phosphorus, making phosphorus models more accurate. So what's the conclusions from this bit? First of all, we were able to narrow down the parameter values, make the models more accurate. We came up with which of the homogenized models was most appropriate. We also in the process found some other unexpected results, that sulfur, for example, could be as a surrogate marker for mycorrhizal hyphae in the mixed phases of soil. But let's move on. The next bit that we looked at was water uptake and how much plant soil interaction influences the water uptake. So we already dealt with hairs. What we found from our imaging is that roots grow into soil and displace the soil particles. So it's easy to intuitively understand that leads to compaction of soil. But what we actually found is that near root surface, there is a higher porosity zone when there's root hairs present there, and then followed by a compaction zone. So we have this sort of dip. And we wanted to know, what does that dip, for example, mean for water uptake? Now, the classic way of modeling plant soil water movement is by using Richard's equation, which combines mass conservation with Darcy flow with two experimentally measured quantities. One is the soil water release curve, that is a capillary pressure saturation relation, that is done in equilibrium, and also experimentally measured hydraulic conductivity as a function of saturation. Now, the problem with those two extra experiments needed for air and water movement in soil is that those are very long experiments. They take a lot of effort. It's also clear that the both water release curve and hydraulic conductivity exhibit hysteresis. So the wetting behavior is different from the drying behavior. And it would need to be measured explicitly for each soil, for each microstructure. And you can imagine that you would need to basically measure it for different soils in different distances from the root surface. Now, there are two problems with this, in a sense that even if we could take the sample from these three different regions and run the classical experiments to measure water release curve, because those experiments take a long time, the soil in the beginning of the measurement wouldn't be the same as it is in the end, because soil microbial processes would change over time. And so we started asking, well, could we not characterize the Richards equation theoretically using the same techniques that we had to multi-scale homogenization techniques? That could we derive them from the images? So what we did, we took the phase field approach, which I'm sure you all in this community are very familiar with, and we applied homogenization theory to it, and then played around with the contact angle. So what plant roots and root hairs in particular are known to do to water in soil pore space is that they change the contact angle. So we can now computationally track the wetting drying behavior in soil and see the Heinz jumps. And what you see is that even little change in contact angle from full wetting to 20 degrees results in drastic changes in this hysteresis behavior in soil. So this was really quite useful thing for us in terms of explaining the science, the impact of the root exudates that do this change of contact angle, how much impact that has on water movement in soil. And it's quite dramatic, as you can see here. We then took those samples and correlated them with X-ray fluorescence and Xane's elemental mapping again. And what we found was basically a curious thing, that because we have these three zones, we have that increased porosity zone near root, followed by that compression zone that was due to the root pushing soil particles aside, and the bulk soil. All of the biological transformations chemically were confined to that increased porosity zone near the root. So what root does by growing into the soil, it basically almost physically seals off with that compression zone the increased porosity sort of bacterial incubator zone near the root from the bulk soil, because in bulk soils we really didn't see much of these biologically dominated transformations, using again the X-ray techniques in the soil. But let's move on, and let's move to the nitrogen studies we have done on a field scale now. So nitrogen is used in 50% of global fertilizer food production. It's really Haber-Bosch is really expensive process for producing fertilizers. It uses lots of energy, but most concerningly, 40% of actual applied fertilizer is actually denitrified back into atmosphere in the form of greenhouse gas. So we started modeling this process, and first we started by looking at the granule dissolution. So what happens if we put the fertilizer granule in a soil? Could it be that it actually has a adverse effect on soil microbiome? So we took a reasonably standard nitrogen model that had the transformations between ammonia nitrate and organic nitrogen between bound and fluid phase reactions. Here the green arrows show the bacterially mediated reactions. We imaged the root, one of the granules in soil, and then predicted, okay, what's the profiles of these chemicals as the fertilizer granule is dissolving? How does it spread its effect into the soil pore space? We then linked these calculations, so the fertilizer granule is in the left-hand side corner here, to the microbial respiration studies. So we know that if the salt concentrations go up, the microbes die. And we describe the death. Our definition of death in this case was that the microbial activity dropped to the 10% of initial to define it as death. And what we found is basically, so it's the same simulation here, top and bottom. It's just in top we plot the death zone, our definition of death. And in the bottom bit, we plot the reduction in activity. And what you see here is that this death zone for microbes is really dependent on the level of water saturation in a pore space. So when it's drier, there's more death for microbes in a soil, whereas in a case of higher saturation, the dilution effect for fertilizer as it dissolves means that there's less death, but there's still activity change. There's still reduction in microbial activity in soil. Now linking this to root system scale modeling, we can start again asking questions. Now I'm cutting short this how we developed the field scale model using our mathematical techniques, but needless to say, we're not doing the X-ray based every single root in soil. We've done the averaging of soil nitrogen uptake. And those codes are quite fast now, so that they allow us to run simulations over many different scenarios very quickly. And we started testing our model against different rainfall patterns, because we suspected that the rainfall pattern from that study of fertilizer granular dissolution, the rainfall pattern must have a strong influence on nitrogen movement and nitrogen uptake in the soil. So we ran lots of simulations. In the UK, the nitrogen is applied twice to the crop during growing season, typically twice. Sometimes it's also applied 3 times. So we did a 2 application study by using 90 different rainfall patterns. And what we get is basically a response curve to how much crop is taking up nitrogen. And having run the simulations for these blue lines are rate of rainfall or sort of rainfall events and the intensity. For all the different pairings of this first application and second application, we get a response curve where the red now means is that that was a perfect pairing to get the high nitrogen uptake. And as it gets the cooler colors, it becomes less effective. Now we can link these studies, so in which basically suggest the optimal times for fertilizing for a given rainfall pattern. We can link this now to our microbial death model. And what we find is that in a dry year, microbial activity inhibition zone due to this couple of surface applications of nitrogen fertilizer reaches really quite deep down into soil, that it isn't just inhibiting the microbial activity on top of the soil, but it also reaches and is a lot stronger in a case of dry year as opposed to wet year. And this now motivates extra new studies. So conclusions from this is dry years, there's a lot more depth around the fertilizer pellets. Wet years, it's easy to achieve nitrogen use efficiency, but the overall uptake is lower. So you need to do potentially some other things to stop the runoff. Dry years are possible to get higher nitrogen use efficiency, but you need to do more precision application to achieve these high yields. This is where I also want to end now, because I think we're running out of time. So why do all these image-based models? Because they are quite labor intensive. They require lots of image analysis skills, lots of analytic skill, mathematical analysis skills, and also computational skills. In my mind, these models are alpha models. They basically allow the most testing to do the most detailed science and testing of the most detailed hypotheses. It also allows those image-based models allow us to validate the upscaling models faster. And that is really important if as mathematicians, we want our models to be used by experimentalists, because some of these techniques, especially homogenization, can be very technical. And they can look a bit like bewilderingly complex to experimentalists. But if you can show the comparison between image-based model and homogenized faster model, and that they are really accurate, then that builds experimentalist use of confidence in models. I also want to, because this is a mathematical audience here, is that I want to encourage all experimentalists or theoreticians to get involved in experiments, because you have a unique insight into the systems, and something that might be obvious to you, you need to communicate it to experimentalists and maybe get your own hands dirty and do the experiments yourself, because it is quite fun and it can stimulate things and make your models better if the more closely you get involved in experiments. So don't be scared of them. But I want to finish here. I obviously didn't do all this work by myself. I have a great team of postdocs and PhD students who are here. I have a great team of collaborators who keep me grounded, who make sure that we are doing the really valuable science that will help they and their experiments. And I want to finish by acknowledging the use of these fabulous synchrotron facilities that we've been very lucky to work with. So Swiss light source, Diamond light source, and Brazilian synchrotron are all fabulous facilities, and you couldn't do anything without funding, which has come from many different sources. So I think I'll stop here.
Thank
Any
you.
questions?
Yes, if there are questions that can be made either just jumping in or in the question and answer. It's an incredible complex problem because I didn't realize, but you need also to take into account the interaction with the rhizosphere. So you have also to model what's happening at the microbic and fungi level. It's really complicated. So also very exciting because there's probably still a lot of open problems to be addressed. Just one question that is more on the use of this analysis. You mentioned the use, the need of a better use of fertilizer, right? So I was in the introduction mentioning also the issues that certainly will arise about climate change. Can your type of analysis also help in understanding how to somehow mitigate the possible effect of climate change on crops?
Yes, so the one way, so soil is the largest carbon storage, terrestrial carbon store in the world, and so locking more soil carbon in the soil is the one way of mitigating the climate. Now, and that is intimately related to these microbial death spots in soil, and also any. So nitrous oxide is the major and very potent greenhouse gas. So if nitrogen isn't taken up by plants, it can get released into the atmosphere and contribute to climate change. So making that fertilization strategy more high precision to make sure that all of the fertilizer that is applied is taken up by plant is the one solid approach taking to mitigate climate change.
Yeah, I was not just only thinking about mitigating climate change, but mitigating the effect, because I suppose that in many parts of the world, the climate change will have a lot of effect on the crops, on the type of plants that one can grow. I think that this kind of study can also give some indication on what to do, right?
Yes, exactly. So precisely, this is why we're doing it. For it, the key always is, and there's carbon storage, the sort of trading that is happening in terms of you don't want to release that carbon that the plants have stored in the soil. One way of doing that is to stop sort of one advice is to stop plowing, for example, of fields. So you have min-till situations. You can use these models in all these different scenarios to test the possible scenarios and possible soil management strategies. Because and where modeling becomes really valuable there is that you can test them out in the computer before you go and do the field trials. Because you want to do only the most impactful field trials where you will actually learn new science about how the system works, rather than repeating known knowledge.
You mentioned reduced order models. Are you planning also? This is just I cannot ask about the new thing that people is doing about using neural networks to build reduced models and things like that. Do you think that it can be applied also in your case?
Yes.
In
So
certain
all
situations they work, in others they work less well. So
yeah, so the AI is a big field, obviously, and the neural networks and machine learning. The problem is they're very data hungry fields. And in a case of when you're dealing with soil, you are in you are a very data poor situation because of the unobservability of soil processes, because it is sort of, it's underground, it's in the, underneath your feet in opaque manner. And so one way we are using AI is that we use a combination of the scarce data that we do have. We then integrate that with modeling, and we run models in terms of sort of generating almost synthetic data sets for training them things. But the AI does depend on the data, what it sees. And there's not enough soil data that is sort of on a profile scale. Basically, there's lots of sort of drones flying over this and that, but that's above ground. But most of this carbon that is relevant for climate change is stored inside the soil.
Okay, so it's
and it's difficult to observe.
Yeah, absolutely. Now that's a problem. Unfortunately, very common in people that deal with porous media. Often you have difficulty in knowing.
Yeah, exactly.
So it was a very exciting talk. I will. We are working on, as you probably know, some of these mixed dimensional models, but not at the level I've seen here. Coupling one-dimensional model for blood flow, for instance, and general model for tissues. So this is also a nice field. There's one of root soil interaction where these models maybe can be of interest.
So you in your introduction you mentioned this that I'd worked in Harvard Medical School. One of the reasons for this was that the root systems, the way they function on extracting root systems, are designed to take up nutrients and water from soil. That's very similar to human lymphatic system, which also functions to remove fluid from tissues. Now the difference, in a sense, is that the most crops are grown in partly saturated soil, so you always have air, water. The exception is rice, obviously, which is grown in rice paddies. And this two fluid situation, air and water, makes the modeling a lot more complex in some sense than human body modeling, because human body is well hydrated. With the only difference is the lung, where we have applied those models to lymphatics and blood vessel flows. The modeling techniques are very similar. It's almost the same. In lung, we do have air pockets in the alveoli. So there you get more of this multi-phase behavior happening, but the modeling techniques are really quite translatable across scales. And in some sense, the problem that challenges the modeling in technique-wise is the plant soil system rather than human body system. There are other challenges in human body system in terms of chemistry.
Yes, certainly.
But doing the both together is the bit that transforms the technology of mathematical modeling.
Yeah. No, but I think in this problem of plant and soil interaction, it looks to me even more complex, because really you have the interaction with other, well, also in the human body, in fact, but here it's very important also the interaction with other organisms, right? With the fungi, with the microbe. I've just read a book about the fungi, and I was really amazed on what they do, collaborating in this symbiotic interaction with plants. It's amazing.
That certainly is the case that there's a lot more need for modelers in the plant soil area. There's lots of groups, large groups, who do biomedical modeling. It's a well-recognized area, soil modeling is sort of catching up in terms of we could do with a lot more people entering this field than making the contribution.